Welcome! Today, we're diving deep into a molecule that's a real double-edged sword in our bodies, histamine. On one hand, it's absolutely essential for our immune system, but on the other hand, it's the main villain behind allergies and some really serious reactions. So we're going to break down exactly how it works, and more importantly, how we can control it.
To really get a handle on this, let's start with a real-world scenario. Meet Ellen. She's 16, and like clockwork, her spring allergies are just awful. So she grabs an over-the-counter drug, diphenhydramine. And yeah, it helps with her running nose, but it also makes her super drowsy and gives her this uncomfortably dry mouth. Her doctor then switches her to a different pill, loratadine, and just like that, her allergy symptoms are gone, but with zero side effects.
And that experience brings us to the core question for today. Why do two different drugs, both designed to do the same thing, fight allergies, have such completely different effects on the body? Well, the answer is all in the pharmacology of histamine.
So here's our game plan. First, we're going to get to know our frenemy, histamine. Then, we'll look at its entire life story, how it's made, where it's kept, and how it gets broken down. After that, we'll explore the different receptors it uses to send messages and the actions those messages cause. Then, we'll get into the weeds of allergies and the much scarier anaphylaxis. And finally, we'll look at the drugs we use to target histamine, which will bring us right back around to Ellen and solve our little mystery.
Okay, let's start from square one. What in the world is histamine? And why are we calling it a frenemy?
Well, histamine is a major player in our body's defense league. It's made by immune cells called basophils and mast cells. Think of them as the first responders on the scene. When there's a threat, like an infection or an injury, histamine gets released and makes our tiniest blood vessels, the capillaries, more leaky. This is actually a good thing. It lets other immune cells rush into the area to fight the problem. But, and here's the frenemy part, that very same action is what causes the redness, swelling, and itching of inflammation and an allergic reaction. And it doesn't stop there. Histamine also has a side gig as a neurotransmitter in the brain, where it helps regulate things like our sleep-wake cycle.
So now that we have an idea of what histamine does, let's look at how the body manages it. You see, to really understand how we block it with drugs, we've got to know its whole life cycle, from birth to retirement, so to speak.
Okay, let's follow the journey. It all begins with a super common amino acid called histidine. An enzyme, histidine decarboxylase, comes along and basically snips a part of the histidine molecule off, turning it into histamine. Pretty straightforward. Now, once histamine has done its job, the body needs a cleanup crew. This happens in one of two main ways. The first path is called ring methylation, where a methyl group gets tacked on to the histamine. This eventually creates a byproduct called methyl-imazole acetic acid. Now that's important because we can actually measure this in the blood as a marker for how much histamine is being released, which can tell us about inflammation levels. The second pathway is oxidative deamination. This one removes an amine group and leads to a totally different byproduct called MAA riboside. So you've got one starting block and two separate finish lines for this molecule.
Now, here's where it gets interesting. The body has two very different ways of handling histamine, which we can think of as two separate pools. First, you have the slow turnover pool. This is in our mast cells and basophils. Here, histamine is stored in big batches, kind of packaged up with heparin, and releasing it is a major event. The whole cell has to degranulate or dump its contents, and it can take weeks to build that supply back up. This is the pool that gets called up for big inflammatory and allergic responses. Then you've got the rapid turnover pool. This is found in places like your stomach lining and in your neurons. Think of this as a just-in-time manufacturing system. Histamine is made and released on demand, as needed, without being stored. This allows for really quick, fine-tuned control of things like stomach acid.
So what actually flips the switch to release histamine from that big, slow pool in the mast cells? Well, the fundamental trigger is a rush of calcium into the cell. This causes something called exocytosis, where the cell literally spits out its histamine granules. And a few things can cause this calcium rush. Our immune system can do it with complement proteins, specifically C3a and C5a. It also happens when an allergen bumps into IgE antibodies that are sitting on the mast cell surface. We'll talk way more about that in a bit. But it's not just the immune system. Some drugs, like morphine and tubiculane, can directly trigger this release too. But we can also stop it. Drugs that increase a messenger molecule called cyclic AMP, or CAMP, inside the cell can hit the brakes on this whole process. Beta agonists, for example, work this way.
All right, we've covered the molecule's life. Now let's see how it actually does its job. Histamine communicates its orders to the body by docking with 4 different types of receptors, which have the very creative names H1, H2, H3, and H4. This table right here is pretty much the Rosetta Stone for histamine pharmacology. Let's walk through it. First up, the H1 receptor. This one is the main troublemaker in allergies and inflammation. You'll find it on smooth muscle and in the brain, and it signals by jacking up the calcium levels inside the cell. Next, the H2 receptor. Its claim to fame is getting your stomach to produce acid, but it's also on your heart muscle. It works by increasing that other messenger we mentioned, cyclic AMP. Then there's the H3 receptor. This one's mostly in the nervous system and acts like a feedback switch. It tells the nerve to stop releasing so much histamine by decreasing cyclic AMP. And finally, the H4 receptor. This one helps fine-tune the immune response and is found on hematopoietic cells, the cells that make all of our blood cells.
And what you're seeing here is the beautiful chaos that happens inside the cell once one of those receptors gets activated. You can see how H1 leads to that calcium spike, while H2 leads to a rise in CAMP. These second messengers, as they're called, set off a whole chain reaction. A key part of this is protein phosphorylation. It's like flipping a bunch of little switches on different proteins, turning them on or off. This whole cascade eventually changes which genes in the cell's DNA are being used, and that's what ultimately changes what the cell is doing. This whole complex process is how a simple molecule can have such huge effects on the body.
So what does all that molecular stuff translate to in the real world? Let's zoom out to the big picture. In your smooth muscles, H1 activation makes them contract. When this happens in the bronchi of your lungs, it causes those asthma-like symptoms. In your cardiovascular system, you've got a two-part story. H2 receptors on the heart speed it up and make it pump harder, while H1 receptors on your blood vessels make them widen by releasing nitric oxide. In the stomach, H2 is the big one. It tells cells to start pumping out stomach acid. And on your skin, H1 activation creates a very famous, specific reaction.
It's called the triple response of Lewis. If you were to inject a tiny bit of histamine into the skin, you'd see three things happen one after the other. First, within just a few seconds, the spot turns red. That's those blood vessels dilating. Second, within about a minute, you get a wheal. That's a raised, swollen bump caused by plasma leaking out of those vessels. And finally, you get a flare, a larger area of redness that spreads out from the center. This is caused by something called an axon reflex, where nearby nerves get triggered and release even more vessel-dilating substances, like CGRP, into the surrounding area.
Okay, and that brings us full circle, right back to Ellen's runny nose. We've laid all the groundwork, so now let's put it together to see what's actually going on when you have an allergic reaction.
So this is what we call an IgE-mediated hypersensitivity reaction. The very first time Ellen was exposed to, say, pollen, her immune system's B cells made a special kind of antibody called IgE. That IgE then went and latched onto the surface of her mast cells, basically arming them and getting them ready for a fight. Now, the next time she breathes in that same pollen, it binds to those IgE antibodies on the mast cells and links them together. That connection is the trigger. It causes the mast cells to degranulate, releasing a massive flood of histamine. And that histamine is what causes all our symptoms, the running nose, the itchy eyes, the whole shebang.
Now, I want you to imagine that exact process, but instead of just happening in her nose, it happens everywhere in her body, all at once. That is anaphylaxis. It's a system-wide, catastrophic degranulation of mast cells, leading to an unbelievable release of histamine. This causes three immediate life-threatening problems. 1, global vasodilation. All your blood vessels get wide and floppy. 2, because of that, you get severe hypotension. Your blood pressure plummets as fluid leaks out of your vessels. And 3, severe bronchoconstriction. The airways in your lungs clamp down and your epiglottis can swell shut, completely blocking your ability to breathe. It's an absolute medical emergency.
Okay, we've definitely seen the dark side of histamine. So now, let's talk about the solutions. What have we developed pharmacologically to fight back against these effects? Broadly speaking, we have three main strategies. Strategy 1, counteract histamine's effects. Basically, fight fire with fire. The best example is adrenaline and an EpiPen for anaphylaxis. Adrenaline hits different receptors, but does the opposite of histamine. It tightens blood vessels and opens up the airways. Strategy 2, prevent the mast cells from releasing histamine in the first place. A drug like sodium chromoglycate does exactly this, which is why it's useful for things like asthma. And strategy 3, the one we're all familiar with, block the histamine receptors themselves. This way, even if histamine is released, it has nowhere to dock and can't cause any trouble. This is the world of antihistamines like diphenhydramine and cimetidine.
So we know blocking receptors is a great plan, but, and this is the whole point, not all antihistamines are the same. Now we're going to break down the different classes, the different generations, and this is where we'll finally crack the case of why Ellen's 2 allergy pills were so different.
This slide has all the answers. Let's start with first-generation drugs. Like Ellen's first pill, diphenhydramine, they're just not very specific. Sure, they block H1 receptors, but they also block other receptors, and that causes all the side effects. They cause sedation,
That dry mouth comes from anticholinergic effects, meaning they block another neurotransmitter, and they can even cause low blood pressure. The reason they make you drowsy is because their chemical structure is neutral, which lets them waltz right across the blood-brain barrier. Now look at the second-generation drugs, like her new one, loratadine. They are way more specific, targeting just the H1 receptor. But the really brilliant part is their structure. At the body's pH, they are ionized, meaning they carry an electric charge. And that charge makes it impossible for them to cross the blood-brain barrier. No entry to the brain means no sedation. This is why they're so much better for people who need to stay awake and function.
This diagram makes it even clearer. Think of that first-generation antihistamine molecule as a clumsy, old-fashioned key. Yeah, it opens the H1 receptor lock, which is great. But it's also shaped just enough to jiggle open other locks, like muscarinic receptors, which gives you the dry mouth, and alpha-adrenergic receptors, which can cause dizziness and low blood pressure. The second-generation drugs are like modern precision-cut keys. They are designed to fit only one lock, the H1 receptor, and they leave all the other ones alone. No off-target effects.
Of course, we also have drugs that target the other histamine receptors. H2 antagonists, like cimetidine, are used for things like peptic ulcers and heartburn. They work by blocking the H2 receptors on the cells in your stomach, which cuts down on acid production. But there is a critical drug interaction you have to know about. Cimetidine messes with a major liver enzyme system called CYP450. This system chews up and gets rid of tons of different drugs. By blocking it, cimetidine can cause the levels of other medications to skyrocket, which can be really dangerous. We also have newer drugs like H3 antagonists. Pitolisant is a good example. It's actually an inverse agonist. It binds to the H3 receptor and does the opposite of histamine, which ends up boosting histamine activity in the brain. This helps promote wakefulness, so it's used to treat narcolepsy.
So there we have it. We solved the mystery of Ellen's medication. Her drowsiness and dry mouth were just classic side effects from an older, non-specific antihistamine that got into her brain. Her new pill is a newer, more specific drug that stays out. This whole journey just goes to show how one simple little molecule can cause such complex problems, but also carry out such vital jobs. And that leaves us with one last big question to think about. If histamine causes everything from a sniffle to a fatal reaction, why on earth do our bodies depend on it so much?